Okay, in the last video we talked about Darwin's theory of natural selection, just an introduction. Now we're going to look at some of the evidence that supports this theory of natural selection. And the first one is fairly obvious. We talked in the last video about how when you spray insects, insecticide, you kill off a lot of individuals. But over time, you're going to select for some individuals within a population that can survive. And they'll not only survive, they'll probably be able to reproduce and pass on that resistance. That gene that gave them that resistance will be passed on to survivors, who will pass it on to more survivors, and we'll see that over time, pesticides lose their effectiveness. Insects become resistant to them. We see that not just with insects, but we see that with drugs, antibiotics. Penicillin was first developed in the 1920s and 30s. It no longer works for a lot of bacterial infections because bacteria have, over time, evolved resistance. In other words, some individuals within the population had some resistance, survived the antibiotic, and reproduced, and passed on that resistance. This picture is from San Francisco. When I taught in San Francisco a couple years ago, I had a student that had this staph infection, and this is the bacteria that they had. The scary thing about it was not that they had this staph infection, it was just sort of a skin infection and it was unpleasant and they were out of school for a couple weeks, but it was a drug-resistant staph infection and there were no known antibiotics to treat it. So you can imagine if the student came to school and exposed a lot of people to it, they all would have had this infection and they would have no way of getting it treated because this staph infection has evolved, this bacteria, over time and is now resistant to all known antibiotics. And that's very scary when that happens because we no longer have treatments for certain bacterial infections. Another example that Darwin noted when he looked at, he was a pigeon breeder, so he bred pigeons, and he also looked at all of the animals and crops that humans bred. And for example, if you start with wild mustard, this is just a weed, it grows all over Eurasia, Europe and Asia, and it's now been introduced to the United States, it grows all over the United States. But from that one wild mustard plant, we've been able to produce broccoli, Brussels sprouts, cauliflower, collard greens, kale, cabbage, probably all the vegetables you hate the most, you can blame on wild mustard. And again, through selecting for different traits, selecting for flowers, we get broccoli, selecting for small lateral leaves, we get Brussels sprouts. Depending on whatever trait we bred for as humans, we developed all these different vegetables from something that looked like this and probably didn't taste very good. If you think broccoli doesn't taste good, imagine eating wild mustard. Probably not very good. But again, artificial selection, we've done this with all kinds of plants and animals. Look at cows and sheep and pigs. They don't look a lot like their relatives in the wild, right? We've selected traits we want in them, not necessarily traits that would serve them best in the wild. So again, if humans can modify plants and animals, then why couldn't nature? Why couldn't predators, prey, parasites also exert that kind of influence on species and cause change? Another thing that we've learned since Darwin is when we look at structures, we tend to see that though they're very different, for example, the wing of a bat, the fin of a whale, the leg of a cat, and the arm of a human, all have very different functions. But when we look at them, and we look at the bones in them, we can see we have similar bones. They're not similar in size, but if we count up the total number of bones, we see that we have about the same number of bones in these forelimbs, and we have them in about the approximate positions. Now, because of natural selection, they've changed size, they've changed shape, and they've changed function, right? But we can trace all of these forelimbs back to a common ancestor that had a front forelimb that looked something like all of these, but again, not like any of them, because natural selection has selected for all these different traits in these different environments with these different species. So homologous structures kind of suggest to us that animals that have these homologous structures must have a common ancestor, must share common ancestry, and that these all didn't just appear kind of randomly in a bunch of different species. Another thing similar to homologous structures is vestigial structures. So a vestige is something from the past. If you look at dolphins, whales, porpoises, and you look at they have a pelvic bone. Well, that pelvic bone was where they once had hind legs. And if you look at their rear fins, you can even see they still have sort of vestigial toes and feet from when they were on land and walking around. Now, these are just fins. They don't need the toes. It doesn't serve them any purpose. But it does suggest that in the past, these marine mammals, like dolphins, whales, and so forth, once lived on land. And if we look at humans, we have something called an appendix. It's a very small sac at the bottom of your small intestines. Every once in a while it'll swell up and get inflamed and it can burst. It doesn't do us any good. But if we look at our other relatives, the primates, they have a large cecum that actually helps them digest fiber. They chew on a lot of roots and fiber, and that's an important digestive adaptation they have. We no longer eat that high fibrous root plant material, so we don't need it, so it's been reduced in size. Other examples of vestigial structures, we could think of our hair. Our hair doesn't really keep us warm, but again, we're a mammal. We're one of this group of furry creatures, but we have sort of vestigial hair. There's a lot of other good examples of vestigial structures in humans. You have little muscles in your ear. You can't really move your ears, but you still have those muscles. So again, vestigial structures suggest things that were once important in the past but are no longer important due to some environmental change, a change in our environment, a change in where we live and what we do. Another thing that's interesting when we look at evolution is when we look at embryos and development. Whether you're a fox, a human, a chicken, a pig, you all start with gill slits. All these organisms have these tiny gill slits. They all have tails, right? Doesn't matter if you're a fish or a salamander or anything. We all kind of start as if we were going to live in the water with tails and gills. As we develop, you can see the tails go away on some species, the gills go away on most species. But what does this suggest? This suggests we still have genes to produce gills. We still have genes to produce webbing between our fingers. Your fingers are webbed until the 5th, 6th, 7th, 8th week of development, and then that webbing goes away. Those genes get turned off. So developing embryos suggests that we have had this need for these genes in the past when we lived in the water, but now we no longer live in the water. We no longer need those genes to be activated, so they get shut off as we develop. So developing embryos, the fact that we start with tails and gills, lose those tails and gills, lose the webbing in our fingers, again, suggests that we once had a very different environment where those genes were important and stayed on, and due to our change in our environment, they're no longer needed. Those genes get shut off through development. So looking at other examples of evolution and relatedness within species, if we look at proteins, we know or we believe that humans are closely related to monkeys. If we look at a hemoglobin protein in a human and compare it to hemoglobin protein in a rhesus monkey, we can see that there's about 8 amino acids different out of 146. So pretty similar. Compare that to a mouse, we have about 27 differences. Compare our hemoglobin to a chicken, we have 45 different amino acids, right? So again, this suggests that we've been evolving farther from those species. We've sort of branched out in the tree of life, and our primates, the monkeys, the apes, orangutans are our closest ancestors, and what we see is we have very similar proteins to those groups of animals, and less so with something like a lamprey, where you can see our hemoglobin and the hemoglobin of a lamprey are not at all related. Which gets to our next slide. So if the proteins are the same, and proteins can show relatedness or unrelatedness, our DNA also can be used to show relatedness. So right now, we used to classify species based on what they looked like, and we'd say, oh, it's got feathers, it flies, it's a bird, it lives in the water, and it's got gills, it's a fish. Well, now we can actually look at the DNA, and we can get a lot more quantitative with how species are related. So for example, humans and chimpanzees are very closely related when we look at our DNA. This is a partial DNA sequence, but if we look at all of our DNA, it's actually about 98% the same between humans and chimps. Okay? When we look at humans and other monkeys, we see that it's much lower. And when we look at things like lemurs, even lower still. So we can kind of put together a phylogenetic map of our relatedness. And in class, we'll do an activity where we look at DNA and compare our DNA with other species to see how closely we're related. So we'll do that in class. But this kind of sums up some of the observations that support Darwin's theory of natural selection.